Good evening, everybody. Good evening, everybody. Well, I hope my screen is visible to everybody and I'm clearly audible to everyone, right? Well, I'm launching the poll for the technical. I'm launching the poll for the technical checkup now. If you can hear me properly and if my screen is visible to you, then click on yes option. If you can hear me properly and if my screen is visible to you, click on yes option. Good evening. Well, great. Okay. I'm assuming that I'm clearly audible and my screen is clearly visible to everyone. Human health and disease, right? And in this human health and disease, we are done with few things. 1 second. Black background, that's correct. Okay. So what have we done in the previous class? We are done with the cell-mediated and humoral mediated immune response, right? And in today's class, we'll continue from there. We'll continue to the humoral mediated immunity, and in that humoral mediated immunity, there were something called that cytotoxic T cell, which will go back and work in the cell mediated immune response. Don't get confused what I told. The plasma B cell and other lymphocytes will be working in the humoral mediated immune response, right? But the cytotoxic T cell, as it is a cell and it is going to kill the infected cell. That's why the cell is taking part in the immune response here, right? That's why this particular immune response will be called as your cell-mediated immune response. Well, out of all the lymphocytes, cell-mediated immune response में एक ही lymphocyte काम करेगा, that is our cytotoxic T cell. Okay, I'm moving to the next slide. Before starting the class, what we need to cover in today's class, that is humoral mediated immune response हो गया है, cell भी हो गया है, antibodies, types of antibodies. Okay, right? Well, here is a question for you people. Which cell conduct cell mediated immune response? इनमें से कौन सा cell cell mediated immune response को conduct करता है? Option A cytotoxic B cell, option B helper T cell, option C plasma B cell, option D cytotoxic T cell. I'm launching poll for the question and I hope you can answer to the question. Perfect. Not exactly cell mediated immune response. The question is asking, Janvi Balari, read the question properly. The question is asking about the cell mediated immune response. The question is asking about the cell mediated immune response. Don't get confused. Only 33% people have chosen their answer. 30 seconds gone. Last 30 seconds remaining. I want rest of the people to answer to the question. Okay, option A and B is not chosen by anyone. All right, so far. Option A and B is not chosen by anyone so far. Let me check. C and D is chosen by people. Okay. Well, I'm ending the poll now. Sharing the result with the people. And the result is like no one have chosen option A and B. Option C and D is chosen by 50% people respectively. I'll be ending the poll here. Sorry, I'll stop sharing, then I'll come to the point. When you are talking about cell-mediated immune response, remember this one, cell-mediated immune response. The question is not asking about humoral mediated. It is asking about cell-mediated, and in the beginning also, I mentioned that in cell-mediated immune response, cell will be working in, cell will be directly involved. Cell जो होगा वो directly involved होगा, और I can say cell involved होगा. 1 second. Cell involved होगा in the process of in the process of immunity, right? Cytotoxic B cell, okay, ऐसा इस नाम का कोई सेल ही नहीं होता. This is wrong. Helper T cell is going to ask for help. It is going to ask for help in the humoral mediated immune response. ये कहाँ पे काम करेगा? Humoral mediated immune response में, right? Well, next is your plasma B cell. It is going to synthesize antibody. ये क्या produce करेगा? It is going to produce antibody. It is going to produce antibody, which is the direct part of the humoral mediated immune response again, right? Next one is your remaining one. ये दोनों भी cancel हो गया. अभी बचा कौन सा है? Cytotoxic T cell. This cytotoxic T cell is directly going to kill the infected cell. Infected cell we can say. It is directly going to kill the infected cell. Well, so that is why option D will be the correct answer. That is your cytotoxic T cell, right? Is that clear to everybody? Exactly. Like this. Well, I'm moving to the next question now. One more question is visible on your screen. Which of the following cell produces antibody? पहला question में ही मैंने आंसर बता दिया है इसका. And now I can assume that like you can correctly answer to the question because इसके पहले वाला question में भी मैंने already आंसर बता दिया है. Perfect, Nandis, एकदम सही. I have launched poll and I want people to answer to the question. Great. 66% people have chosen their answer. I want the rest of the people to answer to the question. Very good. JNB Balari, Nandi is very good. What about JNB Darwar? एकदम सही. JNB Darwar का भी सही है. चलो फिर. We'll be waiting for the YouTube people to answer to the question. Last 30 seconds remaining. 30 seconds gone. Last 30 seconds remaining. Everyone do answer to the question. Everyone answer to the question. Last 10 seconds remaining. Last 10 seconds remaining. Okay. Okay, guys. So I'm ending the poll now, sharing the result with you people. Result is like everyone have chosen option A, no one have chosen option B, C and D. B, C और D को किसी ने भी choose नहीं किया है. B, C and D को किसी ने भी choose नहीं किया है. Okay, let me stop sharing. And that is correct. The correct answer is your plasma B cell. Plasma B cell. Right? It is going to produce antibody. Memories B cell is going to collect memory. It is going to collect memory. It will be collecting memory. Helper T cell is going to ask for help again. I am writing that one. Ask for help. None of the answer तो हो ही नहीं सकता. So option A will be the correct answer to the question. I'm moving to the next slide. That is your cell mediated immune response को देख लो. अभी जो cytotoxic T cell का रोल मैंने कल बताया नहीं था, उसको पहले देख लेते हैं. Well, in the previous class, we are done with the humoral mediated and cell mediated immune response. One more thing that is left in the cell mediated immune response is the role of cytotoxic T cell, right? After the dendritic cell or antigen presenting cell showcased the foreign particle or foreign material to the T lymphocytes. The T lymphocyte planned in a proper way and it divide all the T lymphocytes, all the army into 3 different categories. Helper T cell, cytotoxic T cell, and one is your memory T cell. Helper T cell went for asking help, right? Memory T cell will be collecting memory and information, whereas the cytotoxic T cell का role क्या है? This cytotoxic T cell is going to kill cell. It is going to kill self cell, and the self cell is your infected cell. The self cell is your infected self cell. Is that clear to everybody? Right? So that is what you can see here also. Macrophages, infected cell is there. Okay, everything is happening, and after that what happens, like memory T cell तो अपना निकल गया, memory बना के इधर निकल गया. Okay, that is your antigen MSC complex, that is antigen presenting complex, ये जो बन गया ये इधर निकल गया. Next one is your helper T cell. Macrophages के बाद यहाँ से रिलीज हो गया helper T cell. This helper T cell is going to ask for the help. ये help के लिए चला जाएगा. इसका काम खत्म यहाँ पे. Right? The memory T cell मेमोरी कलेक्ट करेगा. बचा हुआ और एक सेल है जो कि साइटोकाइंस की तरह काम करेगा. व्हाट इट विल डू, इट विल डिवाइड कंटिन्यूअसली. व्हाट इट विल डू, इट विल डिवाइड कंटिन्यूअसली एंड इट विल फॉर्म द नेक्स्ट सेल, दैट इज योर किलर टी सेल, और इट इज आल्सो कॉल्ड एज नेचुरल किलर सेल. इसको नेचुरल किलर सेल भी कहा जाता है. which is going to act on the infected cell, and after acting on the infected cell, the death of the infected cell will happen. So it is killed by this particular cytotoxic T cell. The role of the cytotoxic T cell is to kill infected cell so that there should not be any sign of infection in the body. बॉडी के अंदर किसी भी तरीके का इंफेक्शन का साइन नहीं होगा. Is that clear to everybody? Right? Great. Take a screenshot of this one, then click on yes option so that I will move to the next slide. Yes, बलारी का हो गया. चलो आगे बढ़ते हैं. Next, humoral mediated immune response. I don't think I need to explain this one once again. Very simple. Infected cell आया, B cell के साथ कांटेक्ट में आ गया, T cell वगैरह सब अपना काम कर लिए. Then it come in contact with the B cell. The B cell will be now activation of B cell will be happening, and the B cell will be dividing its army into two categories. First one is your memory B cell, and second one will be your plasma B cell. Memory B cell will be producing more and more memory B cell, and it will try to gather the memory. Whereas the plasma B cell, this is the plasma B cell. Right, the plasma B cell here you can also see. Plasma B cell is going to produce antibody. It is going to produce weapons. That weapons is called as your antibody. Antibody is nothing but your weapon to kill or to weaken the pathogens. Right? Okay, JNB धारवाड़ वाले, मैं प्रीवियस स्लाइड्स में आता हूं. पहले इस स्लाइड का जो भी है ना, इसको ले लो, स्क्रीनशॉट ले लो, फिर मैं प्रीवियस स्लाइड में आऊंगा. ठीक है. Well, I'm going to the previous slide now. This particular slide you want me to show, JNB Dharwad. Check this one. See, this is your antigen MSC complex, multi-histone compatibility complex. This particular MHC means multi-histone compatibility complex. Okay. Right. What is that MSC complex, MSC1, MSC2, many kind of things are there, but that is not like in your level. अपने लेवल में नहीं है वो हायर लेवल में जाकर के पढ़ोगे, ग्रेजुएशंस में और आगे जाकर के. ठीक है. Okay, I hope this is done now. दरवार, is it done? ये स्लाइड हो गया दरवार वाले. JNB दरवार. It done? If it is done, click on yes option. If not done, click on no option. I'll stay in this slide. Great. Well, so I'm moving to the next slide. Now, if you want to make a note of this slide also, take a note. I'll be waiting for you people. Note it down. Note it down. This is your humoral mediated immune response. Well, I hope this is also done, right? If not, click on no option or just let me know in the chat box. Is it done? Darwad, Balari, Nandis. Have you noted this one? Click on yes option if you have noted. Great. Well done. So I'm moving to the next slide now. And in the next slide, you can see how the humoral and cell-mediated immune response can be working, right? First, exposure to the antigen engulfed and displayed by the dendritic cell. That is what I explained also earlier. You will share those slides now. Exactly, Nandis. I'll be sharing these slides. Well, but like I'll be sharing the slides, but those people who are in the school, they don't have mobiles and other things to check for the notes. That's why. Anyway, so first exposure to the antibody. Let us look into this particular thing right now. अभी देख लेते हैं थोड़ा सा. First exposure to antibody हुआ, उसके बाद and जो भी हुआ reaction वगैरह that everything happened, and after that the antigen presenting cells, that is also called as your dendritic cells. Antigen presenting cells, APC, and dendritic cell, it took the material, or it simply what happened, this particular dendritic cell will divide or convert the antigen into many fragments. What it will do, the dendritic cell, suppose this is the dendritic cell, it will engulf the material. Which material? It will engulf the antigen. Suppose this is your antigen, right? और ये अपना जो है ये dendritic cells है. ये तो बताया था मैंने previous class में भी. Right. So what it will do, this dendritic cell is going to divide this particular antigen into many fragments. And after the reaction, how it will look like, the dendritic cell will take all the fragments of the antigen on its body. अपने body surface में सारे fragments को ऐसे ले जाएगा, ऐसे ले लेगा on its body surface. And it will showcase this particular fragments to the lymphocytes. ये किनको दिखाएगा? ये lymphocytes को लेकर के दिखाएगा, बताएगा कि this is the sample of the enemy which has entered into our body. This is the sample of the enemy which is very much dangerous and has already killed many of us. Now, out of all the T cell, the T cell will now like it will divide all the T cell into three different categories: helper T cell, cytotoxic T cell, and memory T cell. Forget about memory T cell. Here we'll be talking about the helper T cell and the cytotoxic T cell. Helper T cell is going to ask for the help. Help के लिए ये जाकर के पूछेगा, किसको पूछेगा? B cell को. And here you can see this is the activation of the helper, like memory helper T cell. I mean like memory T cell. ये बना गया अपना क्या? Memory T cell. इधर अपना cytotoxic T cell अलग से लगा, जो कि किसको मारेगा जाकर के? This is going to give rise again memory T cell as well as your cytotoxic T cell. Cytotoxic T cell बनाएगा और यही cytotoxic T cell is going to defend against the infected cell. मतलब infected cell को ये जाकर के मारेगा, cancerous cells को मारेगा. Okay, and also the transplanted tissue it is going to kill, cytotoxic T cell, right? Whereas the B cell is now again going to divide into two different categories. Its activation will give rise to two different categories. That is what it is going to give rise, that memory B cell and plasma B cell. Memory B cell will be collecting the memory. It will be collecting memory, and plasma B cell is going to produce or secrete antibodies. It is going to produce or secrete antibodies, right? It is going to produce or secrete antibodies. That secreted antibodies will act upon, that secreted antibody will act upon the antigens, and from here the antigen antibody reaction will be starting. So in the next slide we'll be talking about the antigen AZ and antibody AB reaction. Antigen antibody reaction होगा, इसके बारे में हम लोग बात करेंगे नेक्स्ट स्लाइड में, राइट? दिस इज योर सेल मेडिएटेड इम्यून रेस्पॉन्स, जहाँ पे साइटोटॉक्सिक बी सेल काम किया, ह्यूमरल में प्लाज्मा बी सेल काम किया. ओके, Okay, take a screenshot or note the things. After that, click on yes option so that I will move to the next slide. Nandis, JNB Ballari, JNB Dharwad. Nandis का हो गया. What about Dharwad and Ballari? Dharwad and Ballari, is it done? If yes, let me know in the chat box or click on yes option. Dharwad, great. Okay, if you want me to zoom this particular screen to a little, then just a second, huh? One second, don't worry. You can see this one. Is it visible now? Wordings are small. Okay, got it. Well, okay, done then. So, shall I go to the bottom portion of this particular section? Now you can check this one. The other things which you have not written. Okay, then I'll go to the next slide, right? That is activation of B cell. अब इसके बारे में जानने की जरूरत नहीं, ये तो बता दिया मैंने. Okay, I'm moving to the next slide now. Dharwad people, let me know if you want me to stay in that slide. I'll go back to that slide. Okay, well, there is a question for you people now. Question is like, students, check the question. Interference are included in which immunity? Interference are okay. I'll come to this particular question. दरवाड़ वालों को थोड़ा सा नोट करना है अभी भी. अब सही है ना, देख लो. JNB Ballari and Nandis, please wait for a few minutes. Yeah, Nandis, that's correct. Is it done now, Darwar? Let me know in the chat box after you finish the work. Jaldi se. Let me know. Is it done now? Yes or no? ID can on this. Okay. No idea on this. I don't know anything about lockdown or anything. You people are in Karnataka, you must be knowing about that one. Okay then. This is your activation of B cell. Activation of B cell अभी बता दिया, उसको और ज्यादा मतलब बताने की जरूरत नहीं है. Very clear. वायरस अटैक किया, एंटीजन बी सेल जो था वो बी सेल एक्टिव हुआ, एक्टिव होने के बाद lymphoblast form किया. That lymphoblast will be activating the B cell and B cell will be forming two different category of cell, memory B cell and your plasma B cell. Plasma B cell will be producing antibody. Now we are going to talk about the antibody, structure of the antibody, types of antibody, and then your antigen antibody reaction. Is that clear to everyone? Right? So I don't think you need to note this one once again. Okay, so I'm moving to the next slide. Okay, guys, let me know if you want to make a note of this one once again. Well, so here is a question for you people. Question is like, interference are included in which immunity? Okay, interference are included in which immunity? I'm relaunching the poll. Answer me in the poll. Interference are included in which immunity? Option A cytokine barrier, option B physiological barrier, option C cellular barrier, option D none of the above. Interference. What are these interference? Last 40 seconds remaining. I want remaining people to answer to the question. Perfect. Very good, JNB Dharwad. Very good, JNB Balari. Very good, Nandis. Everyone has correctly answered to the question. We'll be waiting for the YouTube people to answer the question. Okay. Last 15 seconds remaining. Last 10 seconds remaining. I'm ending the poll now. Sharing the result with the people. Result is very simple. Everyone have chosen option A, no one have chosen option B, C, and D. So option A will be the correct answer to the question. Interference are the protein. Interference are the protein synthesized by infected cell. Synthesized by virus infected cell. Virus infected cell. Is that clear to everybody? Now I am moving to the next slide. That is your antibody formation produced by plasma B cell, as I told you, and this antibody is a immunoglobulin. It is also called as immunoglobulin. Immunoglobulin. It is also called as immunoglobulin and represented in the form of Ig. Short form में इसको Ig कहा जाता है, immunoglobulin Ig. It is made up of glycoproteins, glucose, or that is your some protein part and some carbohydrate parts. That is your glycoproteins. They are very much specific in nature. And these antibodies are also called as immunoglobulin Ig. Remember this one, right? And they belongs to the group of glycoproteins. If you are talking about antibodies in general, there has been a cartoon, like कुछ कार्टून बनाया गया है, कार्टून शेप इसका बनाया गया है एंटीबॉडी का, जो कि वाई शेप का लगता है, ऐसे, राइट? एंड इसका 2 आर्म ऊपर से रहते हैं, लाइक यू कैन सी इन द कार्टून. ये कार्टून जो तुमको दिख रहा है ना, ये ऐसा इसी तरीके का होता है एंटीबॉडी. You can see this is first arm, this is second arm, this is third arm, and this one will be the outer one, will be the fourth arm. Four arm will be there. Now we'll talk about the structure of the antibody in the next slide. What are the things that are present? If you are talking about antibody, it will be having 2 light chain and 2 heavy chain. These are the heavy chains. H. These are the heavy chains, right? And this will be the light chain. इसके बगल में जो मैं बना रहा हूं, ये light chain होगा. इनका molecular weight light होगा, that is why it is light chain. and इसका molecular weight जो होगा वो heavy होगा, इसीलिए इसको heavy chain कहा जाता है, right? I am moving to the next slide now. If you want to make note, can do it fast. जल्दी से बना लो इसका note. Okay? डोंट वरी, इसके बारे में हम लोग नेक्स्ट स्लाइड में भी देखेंगे, ठीक है? आगे बढ़ता हूं. अभी देखो, ये देखो स्ट्रक्चर ऑफ द एंटीबॉडी का स्ट्रक्चर पूरा बता देता हूं यहाँ पे क्या क्या चीजें हैं. एज आई टोल्ड यू, एंटीबॉडी विल बी फॉर्म्ड बाय 4 चेन. कितना चेन के बने हुए होते हैं? दे आर फॉर्म्ड बाय 4 चेन. 4 चेन का ये बना हुआ रहता है, राइट? 4 polypeptide chain in the sense they are the antibodies are the glycoproteins, in the sense they will be formed of amino acids, and different amino acids will be linked together by peptide bond. That's why it is called as many peptide bond will be there. बहुत सारे amino acid आपस में join किए रहेंगे peptide bond के मदद से, and because of that, that is your many peptide, that is polypeptide, and it is a chain like structure, polypeptide chain like structure. So it is made up of 4 polypeptide chain. Out of 4 polypeptide chain, 2 polypeptide chain will be of heavy nature. It will be heavy. Why it is heavy? Because it is longer, the size is bigger. It is made up of almost approx 450 amino acids, 450 amino acids, and those are identical to one another. दोनों के दोनों एक दूसरे के साथ एकदम बराबर होंगे, identical to one another, right? Okay, is that clear to everybody? Right? And other will be your two light chain. This will be L instead of H. यहाँ पे L होना चाहिए. Okay, two light chain which are made up of 220 amino acids, 450 and 220 amino acids. Okay, these two light chain and heavy chain will be linked together by disulfide bond. You can see in the picture. अभी बता देते हैं, पहले देखो यहाँ जहां पे H लगा हुआ है, H जहां कहीं भी लगा हुआ है, that is heavy chain. ये heavy chain, ये हेवी चेन, मतलब ये जो ब्लू कलर वाला जो लाइन दिखा रहा है आपने को, दिस ब्लू कलर लाइन इज योर हेवी चेन. ये वाला जो है ना, ये अपना हेवी चेन है. और अभी हम लोग यहाँ पे बात करेंगे ये सी का मतलब क्या होता है. ओके, एच फॉर हेवी. सी का मतलब क्या होता है बात करेंगे. एल का मतलब होता है अपना लाइट, ये याद रखो. एल मीन्स लाइट चेन एंड द लाइट चेन इज इंडिकेटिंग दिस पिंक कलर. पिंक कलर का जो है ना उसको इंडिकेट कर रहा है, वो बाहर प्रेजेंट होता है. ओके, दिस हेवी चेन आर identical to one another and they will be connected with one another by the help of disulfide bond, and this region where this is connected with one another by the help of disulfide bond is called as your p-pen cleavage site, or it is also called as your hinge region. It is also called as hinge region. Hinge region भी इसको कहा जाता है. Why? Because this hinge region will be adjusting the size of the antibody. ये जो डाइसल्फाइड बॉन्ड यहाँ पे बना हुआ है, हिंग रीजन है, ये क्या करेगा, एंटीबॉडी का साइज को एडजस्ट करेगा. इट विल एडजस्ट द साइज ऑफ द एंटीबॉडी. एंटीबॉडी का साइज को ये एडजस्ट करता है. ठीक है, 1 सेकेंड, इसको इरेज कर देता है. इट विल एडजस्ट द साइज ऑफ एंटीबॉडी. साइज ऑफ एंटीबॉडी को ये एडजस्ट करता है. राइट, नाउ सी, ये तो हो गया अपना हेवी चेन, लाइट चेन बता दिया मैंने. ठीक है, नाउ व्हाट हैपेंस, देयर आर टू रीजन. वन इज योर सी रीजन एंड अनदर वन विल बी योर वी रीजन. कैन यू सी दैट वन. ये सी रीजन है, ये भी सी रीजन है, ये भी सी रीजन है. ओनली वी रीजन इज दिस पर्टिकुलर पार्ट. ग्रीन कलर का मैं मार्क करता हूं, ये वी रीजन है. एंड वी मीन्स वेरिएबल हियर. Rather than this particular region, in all the parts, the composition of amino acid will be similar or same, we can say. That's why ये हर किसी एक केस में समान होता है, इसलिए इसको कांस्टेंट रीजन कहा जाता है. ये चेंज नहीं होता कभी भी. सी रीजन, ये नीचे का जो सी रीजन है ना, ये कांस्टेंट होते हैं. ओके, only the topmost part will be called as your V region, that is variable region, and it changes due to change in the amino acid content. Right, and why this is changing, because this variable region will be changing because antibody work specifically. This variable region will be very much specific for a specific antigen. So ये इसलिए चेंज होता है क्योंकि ये स्पेसिफिक होता है. It will be very much specific for a specific antigen. That is why. Is that clear to everybody now? Right? And in this variable region, you can find some location where antigen can come and bind. You can find some location where antigen can come and bind. ये green color का मैंने मार्क किया, ये भी देखो, दो ऐसे regions है. That is your antigen binding site. These are the antigen binding site. So in one antibody there are two antigen binding site present, and that antigen binding site is also called as your paratopes. इन्हीं antigen binding sites को किस नाम से जाना जाता है, paratopes भी इनको कहा जाता है. Can you see that one? This particular thing, इसको paratopes भी कहा जाता है. इसको paratopes भी कहा जाता है. Is that clear to everybody? Right? I hope this is clear to everyone now. Once again I am repeating. I'll go to the next slide and in that next slide you can see the things. जब मैं खुद बना के दिखाऊंगा तो अच्छे से समझ आएगा. दरवार, जन भी दरवार वाले हो गया. हाँ, either take a screenshot or note the things. Nandis, I'll come to that point. Light are non-identical because light have this variable region. Light के पास ये variable region होता है, इसीलिए ये non-identical होते हैं. ठीक है, क्योंकि वो variable region की वजह से वो vary करेंगे बार-बार. That is why they are non-identical. Okay. Well, shall I move to the next slide now? Janvi Darwar. Students from JNV Darwar. हो गया तुम लोगों का? I'm not getting any response from JNV Darwar. Yes or no? Let me know. Yeah. If yes, then click on yes option. If no, then click on no option. Okay then. I'll move to the next slide now. Well, that is what I have made it here, but I can make it once again. और एक बार मैं इसको बना के दिखा सकता हूँ. देखो देखो मैं बना के दिखाता हूँ तुमको. Okay, so what I am going to do, I am making the heavy chain in yellow color. ये heavy chain है. This is your heavy chain and light chain in sky color. This will be light chain. Is that clear to everybody? आया समझ में सबको? Now, अगर हेवी चेन की मैं बात करूं तो ये क्या बन गया अपना, ये मैं कलर चेंज करता हूं अगेन, ये अपना हेवी चेन है और ये अपना लाइट चेन है. ठीक है. हाँ ओके श्योर. ठीक है. इज दैट क्लियर टू एवरीबॉडी? नाउ व्हाट हैपेंस, these regions, यहाँ पे अभी मैं इसको थोड़ा थोड़ा डिवाइड कर दूंगा. यहाँ से यहाँ तक, यहाँ से यहाँ तक डिवाइड किया, यहाँ से यहाँ तक, यहाँ से यहाँ तक डिवाइड किया. ओके, इन द सेम वे इसको भी मैं ऐसे डिवाइड कर देता हूँ. ठीक है. दिस पर्टिकुलर रीजन इज कांस्टेंट रीजन. तो ये हेवी चेन का आई एम मेकिंग इट, ये H फॉर हेवी चेन and C for constant region. This is H for heavy chain, C for constant region. This is H for heavy chain, again C for constant region. This is H for a heavy chain and this will be the variable region V. V for variable region. HV में V का मतलब होता है variable region. Why it is variable? Because the amino acid content will be different. ये डिफरेंट होता है इसी की वजह से. राइट. इन द सेम वे लाइट चेन का भी वैसा ही रहेगा. ये कांस्टेंट रीजन होगा ऑफ लाइट चेन. ये लाइट चेन का कांस्टेंट रीजन, ये लाइट चेन का वेरिएबल रीजन. V for variable. So as it is the variable region, why it is varying? Why it is varying? Because in this particular location antigen is going to come and bind. Antigen आकर के यहाँ पे bind करेगा. रेड कलर में उसको मैं मार्क करता हूँ. What is going to bind here? Antigen is going to bind. That is why it is antigen binding site. Exactly, antigen binding sites. Is that clear to everyone? Right. So this antigen binding site is also called as your paratopes. This is also called as your paratopes. Paratopes भी कहा जाता है इसको. आया समझ में सबको? Now this light chain and heavy chain will be connected with one another. वो connection को मैं इधर बना देता हूँ, ग्रीन कलर में बनाता हूँ और व्हाइट में बना देता हूँ ना. So they will be connected with one another by the help of disulfide bond. Right? In the same way इधर भी वही होगा. S S right? So by the help of disulfide bond, they will be connected. Disulfide bond. Okay, and in this particular location, two set of disulfide bond will be there. One set of disulfide bond will be there, right? And this particular location is showing that is the bond or disulfide bond. It will be forming the hinge region. Disulfide bond तो है ही, उसके साथ साथ इस रीजन को क्या कहा जाता है? हिंग रीजन कहा जाता है, which is going to adjust according to the need. हिंग रीजन क्या हो सकता है? ये फ्लेक्सिबल होता है because of the presence of that particular disulfide bond. ये कैसा होता है? ये flexible होता है. These are flexible and that can be adjusted according to the need. Can be adjusted according to need. According to need. Is that clear to everyone? आया समझ में सबको? I hope this is clear to everybody, right? If this is clear to everyone, now click on yes option. If you want to note, then note कर लो इसका, फिर उसके बाद yes option पे click कर दो. Zinbi Darwad, do you want to note the things? If you want, then you can, no issue. So एक in one antibody, how many paratopes are there? Yes, clear. So in one antibody, how many paratopes are there? No, Nandis. What about remaining people? In one antibody, how many paratopes will be there? Exactly, JNB Palari. Not exactly Nandis. Exactly, JNB Darwar. Well, so in one antibody, this is one antibody. Antigen binding site एक ही है इधर, दूसरा एंटीजन बाइंडिंग साइट इधर रहेगा. सो इधर भी अपना एंटीजन बाइंडिंग साइट रहेगा जिसको हम लोग पैराटोप्स बोलते हैं. इधर भी एक पैराटोप्स रहेगा. सो क्वेश्चंस आर आस्क्ड, हैव बीन आस्क्ड सेवरल टाइम, कितने पैराटोप्स होते हैं बोल. ओके, नाउ आई एम मूविंग टू द नेक्स्ट स्लाइड. हियर विल बी टॉकिंग अबाउट द डिफरेंट क्लासेस ऑफ एंटीबॉडी. डिफरेंट क्लासेस ऑफ एंटीबॉडी के बारे में हम लोग बात करेंगे. There are many different classes of antibody, but in general, we are going to talk about the 5 different classes of the antibody. 5 different classes of the antibody. Okay, that is your 5 different classes: IgG, IgE, IgD, and IgM and IgA. Out of all the 5 classes of the antibody, antibody G, antibody E, and antibody D, GED, is your it will show unit structure, means a single structure. ठीक है, it will be having like only 2 paratopes. इनके पास 2 ही paratopes होगा, ये singular structure show करेंगे, right? Remaining, that is your IgM and IgA, like unimerase, or we can simply say this particular structure as your monomer structure. This GED will be monomer. IgG, IgE and IgD, GED, GED will be monomer, जिसके पास 2 पैराटोप्स होंगे. ठीक है. If you are talking about IgM, that will be your pentamerase structure. You can see this is 1, this is 2, this is 3, this is 4, this is 5, and all the 5 monomer are connected with one another by the help of disulfide bond and a joining chain that is called as your J chain. ये क्वेश्चन पूछा गया है, पहले भी बहुत बार पूछा गया है. They are connected by the help of disulfide bond, and most importantly, remember the name J chain. J chain के बारे में याद रखो. Okay, so 5 number of monomers will be present and they are bound together by the help of J chain. ठीक है, joining chain. And the structure, as it is 5 in number, that is why the structure is called as your pentamerase structure. कैसा स्ट्रक्चर मारा जाता है इसको? पेंटामेरस, पेंटामेरस माना जाता है. Next one is your IgA. IgA is a dimeric structure. IgA is a dimeric structure, and here also the joint is formed by two monomer will be joined together, and is it formed by? It is formed by J chain. J chain से ये बनता है, right? Great. Then click on yes option now. If everything have been noted by you, and if all the things are clear to you. Shall I move to the next slide now? Shall I move to the next slide? Great. I'm moving to the next slide. Well, here is a question for you people. Question, students, read the question now. Question is like, number of paratopes present in 4 chain of single antibody. एक सिंगल एंटीबॉडी है जो कि 4 चेन से बना हुआ है. How many paratopes will be there? Okay. Well, I'm launching poll for the question now. देखते हैं कितना लोग कौन-कौन आंसर करते हैं, क्या-क्या आंसर करते हैं. क्वेश्चन अच्छे से पढ़ो फिर आंसर करो, देखते हैं. Great. Exactly Nandis, exactly. JNB Ballari, JNB Dharwad. Great. Okay, 66% people have chosen their answer. I want the rest of the people to answer to the question. Try to answer to the question. Okay. Last 30 seconds remaining, 30 seconds gone. We'll be checking the YouTube people now. Let us check what they are answering, how they are responding. Okay. Last 10 seconds remaining. Last 10 seconds remaining. Okay then. Well, I'm ending the poll now. I need not to share the result because everyone have chosen option B, and that will be the correct answer. Now the question will arise, if a antibody is there which is having 8 chains. How many chains? 8 chains are there. How many number of paratopes will be there? 8 chains के लिए कितना paratopes होगा? एकदम सही, that is 4. Okay, 8 chain के लिए 4 होगा. ठीक है. So well, I am moving to the next slide. चलो आगे बढ़ते हैं. Now we'll be talking about this different class of the immunoglobulin जहां से क्वेश्चन हमेशा बना है और बनेगा भी. देखो, first one is your IgG. It is a monomeric immunoglobulin and it is the most abundant immunoglobulin. Remember this one, GP, gram panchayat. GP, ग्राम पंचायत. हिंदी में ग्राम पंचायत होता है. Okay, well, so IgG will be transferring or have the capability to pass through the placenta. IgG के पास इतना कैपेबिलिटी होता है कि ये किसके ऊपर से पास कर सके, ये प्लासेंटा को क्रॉस कर सके. It cross the placenta and it transform from mother body to the fetus. मदर बॉडी से, मदर से फीटस के अंदर जाता है. मदर से फीटस के अंदर जाता है. ये एक होता है. और एक होता है जो कि मदर बॉडी से बेबी के अंदर जाता है. This is first one which travel from mother body to the fetus, and another one is there which also travel from mother to the baby. We will be talking about that one, don't worry. So IgG is transferring or crossing placenta. Question has been asked for this several times. Which of the following placenta cross, which of the following immunoglobulin cross placenta? That is your IgG. Well, it is monomeric in structure, that is why it will be having 2 paratopes. Produces in secondary immune response. Secondary immune response में ये generally बनता है। ठीक है, ये इन जनरल मतलब अगर देखेंगे तो इंपॉर्टेंट चीज है आईजीजी के बारे में। ओके, आई एम गोइंग टू द नेक्स्ट स्लाइड डायरेक्टली। Most abundant, and remember two things about IgG. Most abundant, and second one, it cross placenta. Question, two question. Okay, I'm moving to the next slide now. Next one is your IgA. It is dimeric. It is dimeric. Okay. Well, so IgA is dimeric. Dimeric in the sense इसका क्या होगा, इसके बीच में J chain present होगा. You can see in the picture. J chain will be there, which will be connecting the two monomeric structure. Okay, this is immunoglobulin A, and in the immunoglobulin A, if you are talking about question क्या बन सकता है, in total immunoglobulin का 15 to 20% ये contribute करेगा. Along with that, as it is dimeric, 4 paratopes will be there. How many paratopes will be there? 4 paratopes will be there, right? It is a secretory immunoglobulin. Secretory immunoglobulin in the sense it is present in secretions of body, like secretions of body like mucus में ये present हो सकता है, tear में ये present हो सकता है. Well, most importantly secretory के केस में देख लो तो ये milk में present होता है, and in milk यानी येलो कलर मटेरियल जो मदर मिल्क होता है ना, फर्स्ट मदर मिल्क, उसको बहुत इंपॉर्टेंट माना जाता है. दैट फर्स्ट मदर मिल्क इज वेरी मच एसेंशियल एंड शुड बी गिवन टू द बेबी, शुड बी गिवन टू द न्यूबॉर्न. द फर्स्ट मदर मिल्क कंटेन सम येलो कलर मटेरियल, सम येलो कलर मटेरियल, इसके अंदर कुछ येलो कलर मटेरियल होता है, एंड दैट येलो कलर मटेरियल इज योर कोलोस्ट्रम. उसी येलो कलर मटेरियल को कहा जाता है कोलोस्ट्रम, राइट? एंड दिस कोलोस्ट्रम हैव द कंटेंट ऑफ इम्यूनोग्लोबुलिन ए, सो दिस इज सेकंड वन, राइट? कोलोस्ट्रम, एग्जैक्टली, दिस इज योर कोलोस्ट्रम. इज दैट क्लियर टू एवरीबॉडी? सो ये क्या बन जाएगा अपना, सेकंड, इसका स्पेलिंग है colostrum. So that is your colostrum, yellow color material, that is your colostrum. ये तो सबको लगभग सबको आता है क्योंकि human health and disease, sorry, this particular thing is also studied in your reproduction, I guess. Colostrum yellow color material होता है क्योंकि first milk में होता है, mother milk में. Okay, and through that milk it passes from the mother to the newborn. So दो चीज याद रखो, first one is your GP, G will be passing through placenta. Another one is AM, timing जो रहता है ना AM. A will be passing through milk, G will be passing through placenta, A will be passing through milk. That's it, GP AM, इतना याद रखो. Okay, okay then fine. So take a screenshot or note the things, then I'll go to the next slide. If you want to note तो नोट बना लो, ठीक है? और अगर हो गया तो फिर जल्दी से मुझे बता दो, यस ऑप्शन पे क्लिक करके मैं आगे बढ़ूंगा. Okay, well, I'm moving to the next slide now. Next one is your IgM. Next will be your IgM. It is present in total, yeah, basic structure will be same, don't worry, basic structure will be same. Okay, so IgM is pentamerous and it is the largest. ये क्वेश्चन बन सकता है यहाँ से, which is the largest antibody? As it is pentamerous, it will be the largest antibody, M. Okay, 7% of total immunoglobulin, and as it is pentamerous, 10 number of isotopes, paratopes will be there. 10 number of paratopes will be there, not isotopes, sorry. Well, it is the first antibody produced during primary immune response. Primary immune response के अंदर जो पहला एंटीबॉडी प्रोड्यूस होता है वो IgM होता है. Serve as a B cell receptor. It means like it will take the information. well serve as a B cell receptor. It means like this particular immunoglobulin M, it will receive the information from B cell as well as what I can say, it will provide information to the B cell also. It will provide the particular material to the B cell also, that is serve as B cell receptor. Okay, now रिमेंबर आईजीएम से क्वेश्चन बनेगा तो वो पेंटामेरिक वाला स्ट्रक्चर बन सकता है. ओके, एंड ये फर्स्ट एंटीबॉडी है जो कि कोई भी इम्यून रिस्पॉन्स में सबसे पहले बनता है. दो क्वेश्चन. इट इज द फर्स्ट एंटीबॉडी प्रोड्यूस्ड ड्यूरिंग प्राइमरी इम्यून रिस्पॉन्स. ओके, नेक्स्ट वन आईजीडी, अगेन मोनोमेरिक होता है. ओके, एंड ये भी बी सेल रिसेप्टर की तरह काम करता है. बी सेल रिसेप्टर की तरह काम करता है. ओके, ओके, बी सेल रिसेप्टर की तरह काम करता है. सो दो बी सेल रिसेप्टर की तरह काम किए, कौन कौन? अच्छा ठीक है, वही चीज यहाँ पे लिखा हुआ था, देखो. जीपी विल बी क्रॉसिंग प्लासेंटा, एएम will be crossing or will be passing through milk, and another one will be your MD or DM, कुछ भी बना दो, डिस्ट्रिक्ट मजिस्ट्रेट, और इसको बता देते हैं हम लोग मेडिसिन, डॉक्टर इन मेडिसिन, MD. So MD are the group that is serving as a B cell receptor. ओके, ये B cell receptor की तरह काम करते हैं. Okay, now let us move to the IgE, the last one. IgE में चल जाते हैं. IgE, that is the last one. Okay, IgE के बारे में बात करें तो ये यहां से क्वेश्चन पूछता है. क्यों पूछा जाता है? क्योंकि IgE जो होता है वो सबसे लीस्ट अमाउंट में होता है. IgE जो होता है वो सबसे लीस्ट अमाउंट में होता है. IgE सबसे लीस्ट अमाउंट में होता है. एंड मोस्ट इंपोर्टेंट थिंग about IgE is it works in the allergic reaction and parasitic infection. Parasitic infection and allergic reaction में ये काम करते हैं, इसीलिए क्वेश्चन बनता है. ओके, I'll go to the slide none this. ठीक है, take a screenshot. चलो ले लो, IgD का ले लो स्क्रीनशॉट. बी सेल रिसेप्टर की तरह काम करता है. ओके, हो गया. Now I am going to the next slide. Here you can see IgE is the one which is the most, or which is the least, which is present in least quantity, monomeric, and it is produced in allergic reaction. Suppose how a doctor will be saying that you have allergic reaction against a particular thing. Suppose you are exposed to dust and you have allergy for the dust. Now you went to the doctor. क्यों क्या हुआ? डस्ट में जाने के बाद तुम्हारा कंटिन्यू स्नीजिंग वगैरह होने लगा, कुछ सिम्टम्स दिखा, स्नीजिंग हुआ डस्ट में एक्सपोज होने से. When you got exposed to the dust, you started sneezing. You went to the doctor and doctor checked everything and he found out that you have, or in your body, he found out that in your body the immunoglobulin E concentration has been hiked up or increased when you come in contact with the dust. From there he confirmed that you have allergy against a dust particle. डस्ट से तुमको एलर्जी है, क्योंकि एलर्जी होने से इम्यूनोग्लोबुलिन ई का कंसेंट्रेशन बढ़ जाता है. ये सीक्रेट होता है. ओके, बात करेंगे एलर्जिक रिएक्शन के बारे में इन दिस चैप्टर आल्सो, इन दिस चैप्टर ओनली एलर्जिक रिएक्शन के बारे में बात करेंगे. दैट्स ऑल. सो दिस इज ऑल फॉर टुडे गाइस. ये उतना ही है. ठीक है, ये ओवरव्यू है. बता दिया गया है कि कहाँ पे कौन क्या करता है. प्लासेंटा को क्रॉस करता है IgG. ए मिल्क के थ्रू चलता है, कोलेस्ट्रॉल से होकर के पास करता है. पेंटामेरिक होता है M, JHN, एंड E जो होता है वो एलर्जी के लिए होता है. Z and M, सॉरी D and M, और G and M. दिस इज D and M, दोनों मिलकर के ये B सेल रिसेप्टर होते हैं. B सेल रिसेप्टर होते हैं. B सेल रिसेप्टर होते हैं. बट M is only pentamerous. Only M is pentamerous. Only M is pentamerous. Okay, so that's all for today. Thank you so much. Thank you everybody. Well, we'll meet again tomorrow. Thank you. ये क्वेश्चन हम लोग कल ही करते हैं, अब छोड़ देते हैं आज के लिए. इन सब चीजों को कल करते हैं, एंटीबॉडी एंटीबॉडी रिएक्शन, एक्टिव इम्यूनिटी, पैसिव इम्यूनिटी, एवरीथिंग विल डू इन द नेक्स्ट क्लास. Thank you everybody. I am launching teacher NPS poll now. Please vote for me in the teacher NPS poll, then we can end the class. Thank you. Please vote for me in the teacher NPS poll, then we can end the class. Thank you.